Welcome students on second lecture on plant growth and development. Okay, in the previous video we have studied what is the definition of growth. Growth means what? It involves some metabolic processes. That definition we have studied in the previous class. Today's lecture will start with the new concept called plant growth is generally indeterminate. Up to this point I have came across. What is indeterminate growth means? Plant growth continue throughout the life of the plant due to presence of meristem. Because of the presence of meristem, the plant growth is a continuous up to the lifespan of the plant. That means not like animal. In the animal, the plant growth only up to the animal growth happens only up to the certain age. But in the plant, the growth is continuous throughout the lifespan of the plant because of the presence of meristem. What is meristem? See here at the tip of the shoot, there is a presence of shoot apical meristem, that is a SAM. And in the intermediate point, here there is a presence of lateral meristem, we can call LM, lateral meristem. And at the tip of the root, there is a presence of RAM, root apical meristem. Because of the activity of these meristems, shoot apical meristem, lateral meristem and root apical meristem, there is a continuous growth in the plant. How? Shoot apical meristem helps to increase length of the shoot, root apical meristem helps to increase the length of the root, and lateral meristem helps for increasing girth of the plant. That means thickness of the plant will going to be increased with the help of lateral meristem. We know the example for lateral meristem: vascular cambium, cork cambium are the example for lateral meristem responsible for secondary growth in the plant. So height increases the primary growth, girth increases the secondary growth, and for this primary growth as well as secondary growth in the plants, meristem will helps. Do you know, in this meristematic cells, at this pointer you can see that pen what I am drawing there. See, meristematic tissue present at the shoot and root tip have capacity to divide and self-perpetuate. They continuously have ability to divide and form new cells. Continuously ability to have divide and form new cells as well as root and shoot tips and at the lateral meristem and self-perpetuate. That means have capacity for long existence of cell division. So that is a self-perpetuate, we can call it is. And plant growth where new cells are always continuously added due to the activity of meristem. At the shoot apical meristem as well as at the root apical meristem, continuously new cells are added there. Because of continuously new cells are added at the shoot and root apical meristem, remember, this growth is called open form of growth. Continuously new cells at the shoot apical meristem as well as root apical meristem as well as lateral meristem, new cells are added. That is why we can call it is an open form of growth. We can call it is an open form of growth in the plants, as new cells continuously at the SAM as well as RAM places as well as LM places, new cells forming and continuously addition takes place. That's why it is open form of growth. Okay, again, generally growth there are two types: primary growth in the plant and secondary growth in the plant. What is primary growth? Primary growth, see at the tip of the root, tip of the shoot as well as tip of the root, we are having shoot apical meristem and we are having root apical meristem. Shoot apical meristem and root apical meristem, because of that continuous elongation of plant axis happens. Here new cells are continuously added, length of the plant will get increased, length of the root also will get increased due to the root apical meristem. The length or elongation of the plant will be happens due to the activity of shoot apical meristem and activity of root apical meristem. Elongation of plant or length of the plant will get increased That growth is referred as primary growth. Primary growth, in other words, we can say height of the plant. Height of plant increase. That is, we can call it as a primary growth. And remember, in this diagram, see here they have given, this is a shoot apical meristem and this is root apical meristem. Here continuously cells are formed and added. Arrow mark they have shown in this direction. Here the cells are added. So elongation of this part will be happens. Elongation of this part will be happens. Because at the tip root apical meristem, at the tip shoot apical meristem, the growth happens, the cell elongate in this region. This region cell will elongate. That arrow mark indicates this side. That means elongation happens this side. So this elongation means height of the plant will get increased. That we can call as a primary growth in the plant. What is meant by secondary growth? After attaining certain height in plants called gymnosperm and dicots, plants, due to the activity of this lateral meristem consisting of cork cambium and vascular cambium, cork cambium. These are the example for lateral meristem. This lateral meristem increase girth of the plant. Girth means width of the plant. The diameter of the plant or width of the plant increase. That growth is referred as secondary growth due to the activity of vascular cambium and cork cambium. We have studied in the anatomy of a flowering plant in that topic. What is vascular cambium? Between the xylem and phloem, whatever the cambium is present, that cambium is called vascular cambium. Are you looking this line? This line, remember, this line is the vascular cambium. Vascular cambium. And in the cortex, this black color line, now I am making it red. See, this is called cork cambium. Because of the activity of cork cambium and vascular cambium, the secondary xylem and secondary phloem is produced inside the plant body and the width of the plant will get increased. So here arrow mark is shown in this manner. That means width of the plant will get increased, shoot as well as root. That kind of growth is called secondary growth in the plants. Diameter or width of the plant will get increased here. So you are looking this, here diameter or width of the plant will get increased. When width or diameter of the plant will increase, that kind of growth we can refer as a secondary growth. And height of the plant when increase, that kind of growth we can call it as a primary growth in the plants. Okay, these are the two types of growth generally happens in the plants. SG means secondary growth, where girth means width only, girth of plant increase. So these are the two types of growths we have studied in the plants. Secondary growth and primary growth in the plants. Secondary growth absent in the monocot plant because in the monocot plants there will be no presence of cambium. That's why secondary growth is absent in monocots. Hope student you have understood. Next point I will go, that is growth is measurable. That concept we have to study here. Suppose at cellular level, if you want to count the growth, what we have to do here? Cellular level growth means what? Cellular level growth. Concentrate here. Cellular level growth means suppose this is a cell. How we can say this cell has undergone the growth? Inside the cell there is a liquid called protoplasm. When quantity of the protoplasm will get increased, we can say the cell has undergone the growth. Increased protoplasm. We can also say it is a growth happen in the plants, in the cell, in the particular cell. When quantity of the protoplasm get increased, this is a cell. In this cell, if protoplasm quantity increase, then we can say the cellular growth. Okay, now increase in protoplasm is difficult to measure directly. Inside the cell, it is very difficult to measure how the protoplasm is get undergone in increase in quantity. The measurement of this protoplasm increase is very difficult. That's why, so the growth is measured by parameters. Which are the parameters? Like increase in fresh weight, increase in dry weight, increase in length, area, volume, cell number. By these are the different parameters we can measure the growth. In that first one, we can see the cell number. See, this is a maize plant. In this maize plant, maize at root apical meristem can produce 17,500 new cells per hour. In the maize, at root apical meristem, this root apical meristem in the root of the maize can produce 17,500 new cells per hour. That is increase in the number of cells. The growth happens, we can say, in the maize plant root. Cell size, say for example watermelon. In the watermelon, increase in the size of the cell 3,50,000 times. 3,50,000 times increase in the size of the watermelon. That is a cell size increase. Then only the watermelon size also will get increased. By that we can measure the cell size increase. The growth of the plant can be measured. Length of the pollen tube. See, length, how by length we can measure the growth. How? See, this is the pollen tube which is growing. This is the pollen tube which is growing under the stigma. So over the stigma, then style. Through style, this pollen tube is growing. The growth of the pollen tube means length of the pollen tube get increased. When length of the pollen get increased, then by the length we can measure the growth. Best example for that, pollen tube growth length increase inside the style. By length we can measure the growth also. Surface area. So this is the dorsiventral leaf, that means dicot leaf it is. Suppose this dicot leaf surface area increased like this. This is a A and this is a A dash. From A to A dash, the surface area of the leaf get increased. That means growth happens in the leaf. So leaf surface area measuring the growth can be determined. By surface area we can measure the growth. By number of increase in the number of cells we can measure the growth. Increase in the size of the cell we can measure the growth. Increase in the length of the pollen tube in the style we can measure the growth. Increase in the surface area of the leaf we can measure the growth. So these are the different parameters. By that we can measure the growth. Growth can also measure by increasing fresh weight, increasing dry weight. So by that also, volume of the cell, by that also we can measure the growth of the particular plant or growth of the particular organ. Okay, this is called growth is measurable. Hope student you understood about growth is measurable point. Next point. Phases of growth. Phases of growth. In the morphology of flowering plant, we have also studied about. There we have studied about the regions of growth in the root or regions of root in the plant. We have studied that one. So concentrate. Regions of root. So that is we have to study LS of root. In that we have studied different regions. Suppose this is a root section, LS. In this LS, we know that here the cells are present. They are continuously dividing cells are there. And over that root cap is present. This is a root cap. Okay, what are the first region of this is initially we can call it as a root cap. Above that, this region is called meristematic region. Or meristematic zone. Above the meristematic zone, cell will undergo increase in its length. When cell will undergo its increase in length, that elongated region is called elongation zone. And after that, cell attain certain maturity, permanent size and shape. That particular region where the cell attain definite size, shape, generally in the root hairs are coming from that region. You know, this particular region is called maturation zone. This is also called as phases of growth. These are also called phases of growth: meristematic phase, elongation phase, and maturation phase. By taking one root example, in the root, these are the different zones are coming in that these phases are there. So first one, meristematic. In the meristematic zone, particularly, how the cells are there? Cells are compactly arranged. Cells are compactly arranged in the meristematic zone without intercellular spaces. Each cell is having large nucleus. Large nucleus is present every cell. Dense amount of what is present? Cytoplasm is present. Dense amount of cytoplasm is present. Large nucleus is present. Compactly arranged cells. Cell wall is a thin made up of cellulose. And cells are continuously dividing. Cells are immature. These are the characteristic features of the meristematic cells that we have already best studied. Maximum detailed parts we have studied in the anatomy of flowering plant. What is this elongation zone cells are how? After the division, these cells will undergo increase in its length. When cell, why it is increased in its length? Because vacuolization happens. Vacuolization means vacuole become larger. Vacuole become larger in every cell, and length of the cell will get increased. And cell wall become a thick here. Little bit cell wall thick. Vacuolization happens. Length of the cell get increased. In this region, what is the name is called elongation region. And in the elongation phase, the cell's length will get increased. In this elongation phase, length of the cell will going to be increased. After that, the cell will undergo the phase, like what is that last phase is called maturation phase, where cell acquire permanent size and shape, and cell wall become a thick. Cell lose their ability of division. Where cell wall become a thick, cell lose their ability of cell division. That particular zone we can call it is the maturation zone. Permanent definite size and shape cell will be acquired. Cell wall thick, cell wall here thick, and cell acquire permanent size and shape. Okay, so here what will happen in elongation? Vacuolation happens. Vacuolation means vacuole quantity will get increased. Vacuolization happens. Okay, so these are the things which are taking place in the different regions of growth or different regions of or zones of plant root. And these are nothing but phases of growth also. Meristematic phase, elongation phase, maturation phase. In these phases, what are the different characteristic features of the cells that we have studied here? Okay. Now concentrate here. That we have studied in the meristematic phase, elongation phase, maturation phase. At the shoot and root apex, at shoot as well as root apex, both SAM as well as RAM. This is a SAM, shoot apical meristem. This is a RAM, root apical meristem, where cells are continuously divided, cells in the rich protoplasm in that area, large nucleus that we have studied now, and a cell wall is a thin cellulosic with plasmodesmata every cell consisting of plasmodesmata. Cell wall thin, cellulosic, every cell made up of plasmodesmata. You know the plasmodesmata. Plasmodesmata is the connection between the two cells. We can call it a plasmodesmata. And in the elongation phase, it is just behind the, that means after the maturation, after the meristematic zone. This is a meristematic zone. After the meristematic, proximal to elongation zone will becomes. In this we have studied vacuolization increase, new cell wall deposition happens, cell wall deposition, little bit cell wall thick here. And last phase, what is the phase called? Maturation phase, where it is above the elongation phase, where cell attain the maximum size, cell wall thick after the, in this phase cell wall become a thick, cell attain maximum size, cell wall become a permanent, cell wall thick, cell acquire permanent size and shape. That particular phase is called maturation phase. Okay, so these are the different phases of growth we have studied now. Now the diagram showing detection of elongation by parallel line technique. A, B, C, D, these are immediately behind the apex of the root. These are most elongated parts. A, B, C, D are the elongated parts of the root or elongation phase showing diagram. Okay, now these are the different phases of growth we have studied now. Now there are growth rates. Which are the growth rates? Arithmetic growth and geometric growth. Like that we have to study two kinds of growth rates. What is this growth rate? Means increased growth per unit time. Increased growth per unit time is called growth rate. Growth rate can be determined by arithmetic and geometrical way. What is arithmetic growth? Arithmetic growth, here in this type of growth, see, this is the diagram which is showing the arithmetic growth. Suppose we can consider this is a parent cell. It is divided into two daughter cells. Say these are the, see, initially before going this cell undergo division, we can consider these are two daughter cell. Before that the parent cell is there. This parent cell, so which is divided into two daughter cell. This is the two daughter, these are the two daughter cells. Among the two daughter cell, one cell continue to divide, other cells remain undivided. This is this cell not dividing. Dividing. So this is dividing. Faint blue cell is dividing, dark blue cell is remain undivided. Out of two daughter cell, one cell is continuously dividing, other cell is remain undivided. Such kind of growth we can call it as a arithmetic growth. Out of two daughter cells, one cell continue to divide. This is the dark blue cell and faint blue cell. Faint blue cell is continue to divide. That's why many daughter cells are formed here. And dark blue cell is remain undivided. So such kind of growth is called arithmetic growth. And arithmetic growth, here if you plot the graph time against the height of the plant, so we get here linear curve of the growth will get here. Which kind of curve will get? Linear curve of the growth will get when we plot the graph time against the height of the plant. Okay, this is what arithmetic growth. Shall I see? I hope student you understood about the arithmetic growth means one daughter cell continue to divide, other daughter cell remain undivided. With respect to this diagram, this faint blue color cell continue to divide and dark blue color cell remain undivided. Okay. Now, say arithmetic growth, how it is mathematically expressed? It is mathematically expressed LT is equal to L0 plus RT. What is LT? LT is a length at time t. Length at time t. L0 length at time 0. And what is R? R is the growth rate or elongation per unit time. This is mathematical expression or mathematical equation for the arithmetic growth rate. What is geometrical growth? Suppose these are the two daughter cells. Both the daughter cells are continuous to divide in this phase. In this kind of growth, both the daughter cells, this daughter cell also divides, this daughter cell also divides. So if both daughter cells are divided, that is referred as the geometrical growth. That means what is the kind of division here? Mitosis only. That is arithmetic also mitosis cell division, geometric also mitosis. But here both daughter cells are continue to divide. That's why it is a geometrical growth. In this type of growth, where initially growth is very slow, that phase of growth is called lag phase. Suddenly there is an increase in the growth maximum, that phase is called exponential phase. And when nutrient supply plant limited, the growth becomes stable or stationary. When nutrient supply is limited, the growth slow down and it is a stationary phase attains. Okay, here if you plot the graph time against the height weight of the plant organ, time against the weight of the plant organ. So here which kind of growth is obtained? Sigmoid growth curve is going to be obtained. Which kind of growth curve is obtained? Sigmoid growth curve is going to be obtained when we plot the time versus the weight of the organ or size of the organ. And having three phases in this geometrical growth: lag, exponential and stationary. Stationary attains when the nutrient supply limited to the plant, stationary phase attains. So listen, this is a zygote. This zygote initially undergo geometrical growth. That means both the daughter cells will undergo mitotic cell division. That means initial phases of growth is geometrical growth. After that, certain cells will undergo arithmetic growth and certain cells will undergo geometrical growth here. The faint blue color cells lose capacity of cell division, and the dark blue color have a capacity of division. This particular diagram showing arithmetic growth. The cells which are dividing, that is the dark color cells here, and the cells which are lose capacity to divide, that is the faint blue color cells. So this diagram showing arithmetic growth, and this is zygote division up to this point showing the geometrical growth, we can say here. Okay, so this particular figure showing both geometrical as well as arithmetic growth. Okay, now next point. So what kind of curve is going to be obtained here? Sigmoid curve. Sigmoid curve is going to be obtained. This is the sigmoid curve in the geometrical growth rate is obtained. Sigmoid curve means having three phases: lag, exponential and stationary phases. Sigmoid curve is a characteristic feature of living organism in natural environment. In natural environment, most of the living organisms showing geometrical growth. Even in plant tissues, organs, and different plant organs like leaf, root, stem, all are showing this kind of growth only. What is the growth? The geometrical growth pattern of growth it is showing. What is the equation for this? W1 is equal to W0 e raised to RT. What is W1? Final size, weight, height, number. Final size in number in terms of weight, height and number. That is W1. W0 initial size at time t, beginning of the period. Initial size W0. What is R? R is the growth rate. Here relative growth rate is taken. T is the time of growth and E is the base of natural logarithm. So this is mathematical expression of or mathematical equation for the what kind of growth we can say geometrical growth. Say for example, this is a leaf. This is a leaf. In this leaf, initial size of the leaf is the W0. After that, this leaf is increased up to this point. This is the W1. And what is R? R is the growth rate and T is the time and E is the base of natural logarithm. So this is what we studied about the mathematical expression for the geometrical growth. Now in NCERT they have given one example of these two leaves. Relative growth. R is the relative growth. It is also measure the ability of plant to produce new plant material. Hence final W1 depends on the initial size W0. What is the meaning of this? This is a plant original size is what is the original size is W0. After that it is obtained final size W1. How W0 become W1? How much this much size of the leaf get increased? Because in this material is going to be added, leaf cells are added. When leaf cells are added or materials are added here, number of leaf cells are added here, what is happening? This length of the or surface area of the leaf will get increased. That means this cell is having, this original plant is having capacity to form new cells. When new cells are added, then W0 become a W1 here. This much of length, area of the leaf will get increased. Okay, so competitive comparison of growth is measured by two ways: absolute growth rate and relative growth rate. Means what? Absolute growth rate and relative growth rate means what? Okay. See, what is absolute growth rate? Suppose this is a leaf A, this is a leaf B. Leaf A, what is the initial size of the leaf A? Leaf A initial size is 5 cm square. After that it will increase from A to A dash up to this A dash. How much increase? 5 cm square become 10 cm square. A dash. This is a B leaf. What is the initial size of the B leaf? Is a 50 cm square. It is increased up to B dash. How much it is increased? It is increased up to B dash 55 cm square. Now what is the absolute growth rate? See, this is initial W0 of this leaf is 5 cm square. W1 of this leaf is 10 cm square. This leaf initial size is W0 50 cm square. This leaf initial size is how much? Final size how much? 55 cm square. So here also how much increased? From 5 to 10. That means how much square size increase? 5 cm square size increase. Here 50 to 55 means how much again increase? 5 cm square size is increased. Both leaf A and leaf B, how much surface area of the leaf increase? 5 cm square surface area of the leaf will get increased. This growth in A and B leaf is an absolute growth. This is which growth? Absolute growth. From 5 to 10 means how much square it is increased? 5 cm square it is increased. From 55 to 55 means 50 to 55 means how much? 5 cm square it is increased. So this growth rate, 5 cm square growth rate is absolute growth rate, we will call this. Okay, what is relative growth rate? Relative growth rate means, remember, here leaf A, 5 becomes a 10 cm square. Leaf B, 50 becomes a 55 cm square. So how much percentage is increased? Leaf A, 5 become a 10 means double, 100% growth of that. And 50 to 55 means how much percent? 10% growth will be happens here. So this 100% growth and 10% growth is called relative growth rate. Is called relative growth rate. See now, this leaf A, how you become a 10 centimeter square? That means double it is increasing 5 centimeter square. That means 100% growth rate is there in the leaf A. And leaf B, 50 becomes 55. That means how much percent growth rate is there? Only 10% growth rate is there. This is what we can say the relative growth rate. What is meant by absolute growth rate and what is meant by relative growth rate? We have studied it. What is absolute? Measurement and comparison of total growth per unit time. Total growth in A leaf and B leaf 5 centimeter square. That is absolute growth rate. Measurement, second relative growth rate. Measurement of growth of given system per unit time expressed on common basis per unit initial parameter. Initial parameter per unit initial parameter. How much growth? How much percentage of growth will be happens? 100% here. 5 become 10 means 100%. 50 becomes 55 means 10% growth. That is relative growth rate. Hope student you have understood about what is absolute growth rate and what is relative growth rate. Okay. Now we have to learn here the different conditions required for the proper growth of the plant. What are the different conditions which are required for the proper growth of the plant? For proper growth of the plant, water is essential. For proper growth of the plant, oxygen is essential. For proper growth of the plant, nutrient essential, temperature essential, light essential, gravity essential. All these factors plays very important role for the growth of the plant. For what purpose water is essential? Water is required for cell enlargement. That means cell has to increase its length. For enlargement of cell, water is essential. Turgidity of the cells also help extension in growth. Turgidity means full of water in the plant cell, which helps for expansion of plant growth. Okay, and water is the medium, provide medium for all enzymatic activities needed for the plant growth. Without water, there is no enzyme plays a role or no enzyme will be active. Water is a medium. In that medium, the enzyme of various biochemical reactions which are taking place in the plant, enzymatic reactions which are taking place in the plants, which are operate in presence of water only. So water is very important role it is playing for cell enlargement as well as turgidity of the cell as well as medium for various enzymatic activities. Oxygen. Oxygen required for metabolic energy for growth. Metabolic energy for growth means what? See, you know the respiration equation C6H12O6. In this 6O2 will be combined and gives rise to 6CO2 plus 6H2O along with 38 molecules of ATP will form. So please tell me, in this reaction, that means this is a glucose which is going to be breakdown in presence of oxygen, and what is going to be released here? Energy is going to be released. That means without oxygen there will be no energy released in aerobic respiration process. If no energy released, then there is no growth of the plant, and various water absorption processes are also not going to be takes place with the help of active transport process. So for the glucose breakdown, oxygen is essential and for production of energy. So oxygen is very important role it is playing in the plants. Without oxygen there will be no release of metabolic energy. Metabolic energy in the respiration, you know that what is the metabolic energy is released? ATP metabolic energy is released with the addition of oxygen molecule. When oxygen is added here, then only the energy is released, and that energy plant utilizes for its growth. Second, nutrients required, that is micro and macronutrients, are required for synthesis of protoplasm and acts as a source of energy. You know that many plant cell, this all protoplasm of all the plant cell made up of micro and macronutrients. Without that, there is a no protoplasm formation. Without that, there will be no enzymatic reactions taking place. So, protoplasm for that micro and macro elements are essential. And you know that source of energy acts as a source of energy means ATP, which are having phosphorus as a nutrient. Phosphorus as a nutrient. So, as a source of energy, without phosphorus, there will be no formation of ATP, and ATP is a source of energy. That's what the meaning of that. So for protoplasm synthesis, micro and macronutrients are essential. Is a source of energy means component of ATP is a phosphorus. So without phosphorus, there is no source of energy. And remember, optimum temperature at which plant growth is maximum. Remember, whenever any reaction in the plants are going to be takes place, only at the optimum temperature. If temperature increase, enzyme become denatured. If enzyme denature, the photosynthesis respiration reaction not takes place properly. Photosynthesis and respiration reactions are not taking place properly if there is a no temperature proper, and deviation from the range of temperature detrimental for its survival. Detrimental for survival means what? If high temperature or low temperature, remember the activities of the plant, metabolic activities of the plants are not going to be takes place properly. At high temperature, remember the enzyme will undergo denaturation and transpiration rate, or we can say the photosynthesis rate in the plants not happens properly. Even in the low temperature also, these reactions, whatever the biochemical reaction which are taking place in the plants, which are not happening properly at low temperature. That is why to perform optimum temperature is very essential to perform all the metabolic reactions in the plant. Light and gravity. How light will affect? Without light there will be no photosynthesis. If light is absent, the rate of photosynthesis is also low. And what is gravity? Gravity determines the direction of shoot and root growth. Gravity determines direction of shoot and root growth. Okay, root growth is towards the gravity and the shoot growth is against the gravity. So gravity also affects the growth of the plant. Without light, there will be no photosynthesis. If light is absent, photosynthesis is also low. So these are the various factors which are responsible for the growth of the plants. Without these factors, plant growth not taking place properly. You know, water is a raw material for photosynthesis. You know that equation of photosynthesis. 6 CO2 combines with 12 molecules of water. Gives rise to C6H12O6 6H2O plus 6O2. That means raw material for the photosynthesis what is? Water. Without water there will be no photosynthesis. Without water enzymatic reactions are not taking place properly. For enzyme proper action, photosynthesis reactions as well as plant turgidity maintenance, water is plays very important role. Okay, what is the next concept we have to study here? We have to study the next concept called differentiation, de-differentiation, and what is meant by re-differentiation process. Okay. First we can learn about differentiation means what. Secondly we can learn about de-differentiation means what. And thirdly we can learn about re-differentiation means what. First we can learn about differentiation process. In this process, in which meristematic root cells, that is root apical meristem and cambium, differentiate and form mature specific function. mature specific function. So this is a meristematic cell which will undergo division and form root cell or leaf cell or phloem sieve cells. These are the permanent cells, root cell, leaf cell, sieve tube of the phloem. So from that, the meristematic cells, these are the permanent mature cells are formed. Root cells for water absorption, leaf cell for photosynthesis, and phloem sieve tubes for water transportation. This process is called differentiation process. What is the process? We can call it is differentiation process in which meristem divides. That is what meristem? Root apical meristem, shoot apical meristem, cambium divide and form a mature tissue like root cell, like leaf cell, like phloem sieve tube to perform a different function. That process is called differentiation process. In the differentiation, cell wall protoplasm undergoes structural changes. Capacity of cell division is lost. This cell wall and protoplasm and cell wall. This is a cell wall and this is a liquid. What is the liquid? Is called protoplasm. Protoplasm and cell wall undergo change and cell become a mature. Even cell, when it will form a root cell or it will form a leaf cell, lose cell division ability. Lose cell division ability. Cell division ability. So that is the differentiation process. Best example for that, loss of protoplasm to form tracheary elements. Suppose this is a xylem tissue. In this xylem tissue, we are having, this is a xylem vessel. Xylem vessel or xylem trachea, we can say this. In this xylem trachea, initially what is present? Initially there is a presence of protoplasm. Initially there will be presence of protoplasm. So what is happening during its differentiation process? It will lose the protoplasm. Lose protoplasm. When it will lose protoplasm, its lumen is empty. Its lumen is empty. Its cell wall is having thick deposition of lignin. Cell wall is having thick deposition of lignin. And this trachea of xylem lose protoplasm during differentiation process. This is the process I am showing, the process called differentiation. During differentiation, what is happening here? Protoplasm will losing. When protoplasm losing, cell wall deposition lignin happens, and this xylem trachea transport water from root to leaf, root to leaf or root to sheep, root to shoot. This xylem trachea helps for transportation of water. So this is what the example for differentiation process. Xylem tracheary elements loses the protoplasm, and that lumen of the xylem used for transportation of water from roots to the leaf part. From roots to the leaf part, this xylem vessel loses the protoplasm. Protoplasm lose in the xylem vessel, and water transport from root to the leaf by deposition of lignin and on the cell wall of the xylem trachea. That is example for the differentiation process. Differentiation process. So what is this differentiation means? What we have studied here. What is the de-differentiation process? Under certain condition, differentiated cells regain the division ability is called de-differentiation. That means what? We have studied the secondary growth in the plants. You know, this cambium is called a fascicular cambium. This red color what I am showing with the help of pen, this cambium is called fascicular cambium. Okay, this is a fascicular cambium, and this is the primary xylem, primary phloem. In between that fascicular cambium is there. And between this vascular bundle, this is a vascular bundle, this is a vascular bundle. Here, permanent cells, which cells are present? Parenchyma. Parenchyma is a differentiated cell or permanent cells. Parenchyma differentiated cell. From that, the new cambium is formed here. From parenchyma, it's a differentiated cell. It's a permanent cell. See, this is the parenchyma is a differentiated cell. From permanent or differentiated cell, new cambium is formed here. That cambium is called interfascicular cambium. So this cambium formation process is by de-differentiation process. By which process? De-differentiation process means what? Formation of interfascicular cambium in between the vascular bundle where differentiated which cells are present? Parenchyma present. Here parenchyma are present, and from parenchyma here there will be formation of this kind of cambium, interfascicular cambium. Interfascicular cambium formation in between the vascular bundle from parenchyma. Parenchyma is the differentiated tissue. From differentiated tissue, interfascicular cambium formation is an example for de-differentiation. And why interfascicular cambium is formed? For secondary growth. Inter and fascicular both joining together and forming vascular cambium. Inter and this fascicular cambium joining together and forming vascular cambium, and this vascular cambium helps to increase width of the plant or diameter of the plant, girth of the plant or size of the plant. We can say secondary growth in the plants. So this is called, under certain conditions there will be formation of a new cambium between the vascular bundle. This is the vascular bundle. This is the vascular bundle. Listen here. What we can say here? Here the cambium ring continuously formed. You are looking here. This is the continuously formation of cambium ring, vascular cambium. So concentrate here. So here the cambium ring, the green color line what we are showing is because of inter and intrafascicular cambium joining. Okay, inter and intrafascicular cambium. This is the interfascicular cambium and this is the fascicular cambium. Inter is formed by parenchyma cells by what process? De-differentiation process. By what process? De-differentiation process. So hope student you understood what is de-differentiation between the vascular bundle. This is vascular bundle 1, this is vascular bundle 1, this is vascular bundle 2. Between that parenchyma cells are there. From that this is a new cambium formation takes place. This cambium is by which process? De-differentiation. Which is that cambium? Interfascicular cambium formation during secondary growth in the plants. Okay. What is a redifferentiation process? Remember, what is the differentiation we have studied? What is the de-differentiation means we have studied? Now what is the de-differentiation? Redifferentiation means what we have to study? De-differentiated cells can produce cells that again lose capacity to divide but mature to form specific function. That means the de-differentiated cells again lose capacity to divide. Differentiated cells form permanent tissue. From permanent tissue again regain the division of ability, de-differentiation. De-differentiated cell again losing the capacity of cell division. What is the process is called? Redifferentiation. See, from differentiation, de-differentiated cells are formed. De-differentiated cell lose the capacity of cell division. Once again, when de-differentiated cells lose the capacity of cell division, once again, what is the process is called? Redifferentiation. Redifferentiation. For best example, I will tell you. Concentrate here. Differentiation means what? Simple words we can learn. Meristematic cells become a permanent cells. Become a permanent cell. That means lose capacity to divide. This process is called differentiation process. Okay. Here, now this is the differentiated cells. Differentiated cells that means which cells? Differentiated cells means which cells? Permanent cell. Permanent cells regain capacity to divide. Differentiated cells or permanent cells regain capacity to divide, called de-differentiation. De-differentiation. Again, now what is happening next? De-differentiated cells again lose cell division capacity. This is called redifferentiation. This process is called redifferentiation process. Redifferentiation process means de-differentiated cells again lose cell division capacity. This is called redifferentiation. Remember here, meristematic cell lose cell division capacity, become permanent, called differentiation. Differentiated cells means this only, permanent cells. Differentiated cells means which cells? Permanent cells. Permanent cell again undergo regaining capacity to divide. This process is called de-differentiated. And what is the de-differentiated cells? Again lose capacity to divide, called redifferentiation. This is the process called the redifferentiation. And de-differentiation means what? So redifferentiation, plant growth is open. We know that it can be indeterminate or determinate. Differentiation is also open because tissue cells arising from the same meristem by have different structures at maturity. Means what is the meaning of this? Differentiation is open because from one cell different mature cells are formed. So from the one differentiated cell, you know that root cells are formed, shoot cells are formed, leaf cells are formed, root cells, shoot, stem will form from this, and leaf will form this. From one cell many cells are arises or many structures are arises from the same meristem. At maturity, many cells are arising from one cell. At maturity, each cell become different. This will become a root cell, this will become a stem, this will become a root, a leaf. That means this is an open form of growth. Differentiation is an open form because from the same meristem, different organs are forming. Root, stem, leaf, from same meristem it is forming. That's why it is an open form. Differentiation is an open form. Final structure at maturity or tissue is also determined by location of cell. Final structure of any organ Final structure of any organ or any tissue is determined by location of the cell, where the cell is located, whether it's the shoot tip or whether it's the root tip. That location of the cell determines the final structure of the plant. Shoot apical meristem, form shoot, leaf, like that. Root apical meristem, form root, like that. So where the meristematic cell is present, that location determines the, what we can say, the organ of the plant. For example, root apical meristem differentiated into root cap cells, while those pushed to periphery forming epidermis. See, this is a root apical meristem. Root apical meristem forming what cap? Root cap. Root cap. Root apical meristem forming root cap. And while pushed to periphery, root apical meristem pushed periphery as well as forming epidermis. At the periphery, root apical meristem pushed and forming epidermis. Okay. So these are the three processes we have studied. What is the differentiation? What is the redifferentiation? We have studied. Okay. So I will repeat here. What is the differentiation process? Permanent meristematic tissue forming the permanent cell, that is a root apical meristem or shoot apical meristem, and cambium forming cambium, root apical meristem, shoot apical meristem. Both are, all are meristematic tissue only. Meristematic cells, what are coming? SAM is coming, RAM is coming, as well as cambium is coming. All are meristematic cells. So RAM is forming root cell, SAM is forming leaf cell, and also forming, SAM is also forming phloem tube. So they are forming specific permanent tissue and perform specific function, that is called differentiation. Best example I have given here, xylem trachea loses the protoplasm and helps for transportation of water by deposition of lignin on that. That is the example for differentiation. What is that? De-differentiation means what? Differentiated cells. Say for example, this is vascular bundle 1, vascular bundle 2. Between that, parenchyma cells are differentiated cells. So differentiated cells again regain the capacity of cell division. Regain the capacity of cell division is called de-differentiation. So which cambium formation? Interfascicular cambium and cork cambium formation during secondary growth. Between vascular bundle, this is vascular bundle 2 and this is 3 and 4 vascular bundle we can form. Between that, interfascicular cambium formation and cork cambium formation in the cortex, that is the process called de-differentiation. Divided cells again regain ability. See, permanent cell regain the ability of cell division. Parenchyma is the permanent tissue which regain the ability of cell division again here. That process is called de-differentiation. What is redifferentiation? Differentiated cell, suppose this is a differentiated cell. Differentiated cell again lose capacity of cell division. Become a mature and perform specific function. That is called redifferentiation. This is a differentiated cell. Differentiated cell again loses. See here, de-differentiated cell. What I am telling? De-differentiated cell loses capacity of cell division. Lose cell division capacity. De-differentiated cell lose capacity of cell division is called redifferentiation. Okay, I think hope student you understood what is the differentiation, what is the de-differentiation and what is redifferentiation. In short, meristematic become permanent, differentiation. Okay, and permanent become again meristem, de-differentiation. De-differentiated cell again lose capacity of cell division, redifferentiation. Hope student you understood up to this point. Next concept I will start in the next video.